Hello, grade 12 students. In this video, we're going to talk about our new chapter, chapter 16, birth control. Start with the first document talking about contraceptive methods. First, let's talk about paragraph 1. Paragraph 1 talking about prevention of fertilization. As you know, there are several methods for the prevention of fertilization. Mostly, they are first the male condom. So the male condom is a sheath of latex rubber. It's placed over the male copulatory organ, which is the penis, and this one is used in order to prevent the semen entering into the vagina, shown in figure 1. This is called the male condom. This is the first way for the prevention of fertilization. Second one is called the vasectomy. It's a surgical procedure that involves the severing of the vas deferens. So, by this way, you can prevent the passage of the sperm cells from the testes toward the penis. This is the second way. Number 3, it's called the diaphragm. Diaphragm is the shallow cup of plastic as shown here in figure number 3. It's used in or it's placed inside the vagina and it covers the external portion of the cervix, thus it prevents the passage of the sperms through it. Next we have tubal ligation, number 4. It's a method where the oviducts or the fallopian tubes, they are blocked using a surgery in order to make them incapable of transporting what? The ova. So they prevent the access of the sperm cells. There are other methods like cervical caps, vaginal sponge contraceptives, all of which they are barriers that prevent the passage of the sperms throughout the cervix. These, all of them, are related to what? They are related to the inhibition of ovulation. However, oral contraceptives, also they are known as the pill, are the most widely used by couples to prevent pregnancy. There are two major types of pills. We have the combination pill and we have the mini pill. Combination pill, it contains both estrogen like ethinyl estradiol and a derivative of progesterone, while the mini pill, it contains low dose of progesterone hormone. So here the action of these pills is the prevention of the process of ovulation. So the combination of both estrogen and progesterone, it suppresses the production and the release of the gonadotropic releasing hormone GnRH by the hypothalamus. So the result is the inhibition of the synthesis of LH and FSH, while the mini pill, it contains only progesterone. Thus, it makes the cervical mucus hostile to the passage of the sperm cells and it disrupts the transfer and the implantation of the early embryo. Here are the different types of pills, mini pill and the combination pill as well. You have to know that effect of the pills on the plasma level of the ovarian and the pituitary hormones. You have to know that we have two women. They have null levels of the progesterone in the follicular phase compared to a normal cycle. So the graph of the woman taking contraceptive pill, it shows constantly null level of progesterone and low level of LH and FSH. Here, the prevention of implantation. For the prevention of implantation, the most common method used is the IUD. IUD, it's called the intrauterine device. These are some of their shapes. It's called IUD. This consists of a coil of a flexible plastic. This passed through the cervical canal into the uterine cavity. It's a foreign body. That's why the woman's body will make a response against it to eliminate it. Thus, the IUD, the intrauterine device, will evoke the release of large number of phagocytic white blood cells. So therefore, it will maintain the endometrium in a chronic state of low inflammation. So the increased number of these phagocytic destructive cells will make the endometrium no longer an acceptable place for implantation. These are the different mechanical barriers to gamete fusion among the different contraceptive methods, like the condom. It's used by males. It's to prevent the semen entry inside the vagina of the female. Also, we have the vasectomy, also for the male. It prevents the passage of the sperm cells from testes to the penis. Also, we have the diaphragm, which is the female one use it and prevents the passage of the sperms through the cervix. We have tubal ligation, also for female. It blocks the oviduct, as such denying the spermatozoa access. And we have the coitus interruptus. It's used for both male and female, and it's used as a natural receptive method that prevents the release of sperm inside the vagina, and all of them, they are contraceptive methods. Here we move to document 2, talking about the contrastive methods. One of the most widely used contrastive methods is the RU486 pill, and we have what's known by this technique. It's the mechanical method of the embryonic section, and we have also the destruction of the embryo, and this is are the contrastive methods. Let's step by step. Contraceptive methods refer to any method that's used to interrupt pregnancy and to induce the abortion of the embryo. So, what are the methods based on? Let's start first with the mechanical method of the embryonic suction. They use curettes or they use a suction pump, and one of the methods used to terminate pregnancy is dilation, followed by curettage. In this procedure, the cervix is dilated and the cannula is passed into the uterine cavity. Then it's attached to an electric suction pump that the embryo will be removed by the uterus by suction, by the suction, and the risk of maternal death due to induced abortion increases as the pregnancy advances. So, for medical reasons, induced abortion during the first trimester of pregnancy up to 12th week is relatively safe. So, the first method is the embryonic suction. Second method is the one that's called the abortion pill, RU486. Another contraceptive method is the pill, RU486, which was introduced first in 1980. This abortion pill consists of a chemical molecule called mifepristone and known as RU486 for Russell and Ockliff, research number 486. It's an oral pill that should be followed by intramuscular injection of prostaglandin in order to stimulate the uterine contraction and help for the abortion to occur. In the first experiment to a young female rabbit, we inject estradiol so their uterus becomes sensitive to the effect of progesterone. A few days later, the experiment was performed as shown in document C. So this document C illustrating the contraceptive action of the RU486, where the injection of estradiol in the first experiment into the female rabbit, it induces a weak development of the uterine endometrium, as shown here. However, in the second experiment, due to the injection of estradiol associated with the injection of progesterone, for both estradiol and progesterone was injected, so the endometrium appears like in the form of uterine lace. So we have significant development. While the injection of estradiol and taking RU486 in the third experiment, it gave the same result shown in the first experiment. So RU486 doesn't modify the effect of estradiol on the endometrium. However, in the fourth case, we notice that we have injection of estradiol first, then injection of progesterone, then RU486. So we have the effect, or we notice that the effect of progesterone associated with estradiol is more distinct when the dose of RU486 is smaller. So 1 milligram of RU486, we have higher development than the case of 5 milligram than the case of 20 milligram. So as the amount or as the dose of the RU486 is smaller, like 1 milligram per kilogram, so we have a significant development. From here, we can know that RU486, it has an anti-progesterone effect. It competes with this progesterone since they have a similar structure. We're going to talk about them in the figure D. So RU486, it prevents the implantation and also gestation. Here we can see that RU486 and progesterone in figure D, they are highly similar in the spatial configuration. They are similar in structure, so they can bind to the same receptor. That's why the RU486 can compete the progesterone and can take the opposite effect and can compete it for the function of progesterone and can lead to the abortion as the dose increases. Now we move to document 3, medically assisted procreation. Let's talk about the first one, artificial insemination. Before we start with artificial insemination, you have to know that couples who use contraceptives wish to avoid pregnancy. In contrast, other couples, they wish to have children but are unable to conceive. Either the male partner or the female may be infertile and sometimes both of them. So what are the techniques? One of them is artificial insemination, where a technique where sperm are delivered directly into the uterus by means other than coitus, insertion of the penis inside the vagina. In artificial insemination, AI, the sperm cell they can come from the husband, so we call it AIH, artificial insemination husband, or from a donor, AID, artificial insemination donor. Artificial insemination from the husband is helpful mainly when cervical factors prevent fertilization, or when the male has low count of the sperms. Also, the anyone who is contemplating the AIDS as a means to alleviate infertility problems should be aware of the risks of disease transmission. Both AIH and AID, the main deposit semen into tube and sperm cells, are concentrated with gentle centrifugation, and then the sperm cells are separated from the seminal fluid and resuspended in a physiological solution. Then the spermatozoa are deposited via a cannula high in the uterine cavity close to the oviduct and should be performed under medical supervision. This is the technique of the artificial insemination. You took the concentrated semen using a syringe, and 24 hours after ovulation, it increases the chance and transfer. Cervical insemination, the sperm cell will transfer it, and in vivo, inside the body, fertilization occur. So stages of artificial insemination: first, we have to stimulate the ovaries with hormones. Second, ultrasound control the size and the number of the follicles. Number 3, we have the release of the egg from the follicle. Intake of the hormones stimulate this process. Preparation of the semen sample, and the husband must pass sperm sample, and then the expert will select the highest quality sperm, and then insemination. Step number 5, on the day of ovulation, take place where selected sperm cells they are injected in the human uterus using the thin catheter, this one like a syringe. IVF and ET, in vitro fertilization and embryo transfer, where in vitro fertilization and embryo transfer are devised mainly for women with a blockage inside the oviduct that prevent the passage of the gametes, sperm cells, and eggs, and hence it interferes with fertilization. For this technique, the woman will receive FSH injections to stimulate the growth and maturation of multiple follicles in the woman's ovary, superovulation. Then oocytes are removed by suction from large follicles in the ovary before the ovulation. This requires a simple surgical procedure that includes the insertion of a cannula in the follicle through the abdominal wall. The ova are then examined in the laboratory or incubated in vitro inside the lab under proper condition with the sperm cells to allow the fertilization process. IVF embryos are allowed to reach about 8 to 16 cells in vitro inside the lab. This requires about 3 to 4, even 5 days. 3 to 5 embryos are then placed in a tiny tube and then inserted in the woman's uterus through the vagina. Finally, the embryos are released into the uterus. Several embryos are transferred to increase the chance of the pregnancy process. So, step number 1, stimulation of the follicle. Step number 2, ovarian punction and follicular aspiration. Step number 3, we have implantation, we have oocytes, we have semen preparation and fertilization in vitro. The cleavage and the embryo, they are transferred. So this is the ET, embryo transfer, and all this process called IVF, in vitro fertilization. So this is the in vitro fertilization, where the sperm cells and the egg cells, they are fused inside the lab in vitro in order to allow fertilization, and the fertilized egg are then introduced into the uterus for embryo transfer process.